and we're being recorded, and the impact on post-traumatic stress disorder, substance abuse, and violence. And this covers over 12,000 participants from urban Atlanta. Dr. Ressler is previously a Howard Hughes investigator, and he's also a member of the National Academy of Medicine. His work, as you see, bridges broadly translational research from molecular neurobiology and animal models to human genetics and epigenetic research on topics of emotion, particularly fear and anxiety disorders. He has published over 300 manuscripts ranging from the genetic basis of molecular mechanisms of fear processing to understanding how emotion is encoded in the brain across animal models and humans. We welcome you, Carrie. Thank you for giving our keynote and getting us started. We're looking forward to learning from you.
Thanks so much, Hadeen. It's a delight to be here today. The work at the Connor Center is just fantastic, and I really look forward to future collaborations and learning more, and I'm very excited to be able to share a little bit of our work today with the Connor Center Symposium. The title of my talk today is Sex Differences in Post-Traumatic Stress Disorder and Other Stress-Related Disorders from Cells to Communities. Next slide, please. So I'll be focusing really on the concept of trauma. And this slide from Sandra Galea, who's now Dean of the BU School of Public Health, formerly of Columbia Public Health, really illustrates the role of trauma across all of our disorders, both in terms of morbidity, mortality, and cost. So this shows that combined civilian and institutional active duty military mental health disorders are the top cost in medical spending over the last decade. And trauma, while physically shown here on slide 3, we know is one of the biggest risk factors for almost all psychiatric disorders, especially if you consider early childhood trauma and others. So focusing on post-traumatic stress disorder in this slide shows as really the best well understood disorder combining both the mental health effects of trauma exposure and our best understood stress disorder. PTSD occurs in about 5 to 10% of the general population, so it's already quite significant. However, if you include and focus on those most at risk, so combat veterans and at-risk populations, and I'll be talking about some of our work in inner cities in America, you even see 2 or 3 times that rate, up to 25%. I'll talk a little bit more about why PTSD, in addition to its clinical significance, is important because we actually know when it starts, and it's one of the few psychiatric disorders that we may actually be able to intervene and prevent. Just to put us all on the same level playing field, PTSD involves 5 components. First, the event. Person has to have been exposed to death, threatened death, actual or threatened serious injury, or threatened or actual sexual violence. And that last was added with DSM-5, particularly representing how frequent PTSD is with interpersonal assault and rape. From a neurobiological perspective, it's so important to understand the intense fear and helplessness that's experienced by the victim of a trauma. PTSD, after 1 month of ongoing experiences of the following symptoms, can be diagnosed. And those include intrusive symptoms, re-experiencing of the traumatic events such as nightmares and flashbacks, avoidance symptoms, avoidance of the reminders, the stimuli that reminded one of the initial trauma cue, and negative alterations in cognition, difficulty focusing, difficulty thinking, as well as depressive symptoms and anhedonia and numbing. And finally, some of the most specific of PTSD is sympathetic hyperactivity, the decreased sleep, the increased startle response, hypervigilance. All of these can be really thought of as a person who is constantly on edge as if they are still reliving the trauma, the life-threatening situation, and they can't get it out of their head, if you will. Next slide, please. We often think about PTSD across sexes. However, we know that across many different studies, and I'll show another slide in a minute, but that women have about a 2-to-1 female to male ratio for PTSD. And this is even after we control for a type of trauma, for example, sexual trauma and age of exposure. The top graph shows a large national mental health study from Ron Kessler, and the bottom graph shows some of the data I'll show you in emergency departments, in which both looking at rates of PTSD across ages, we see about a 2-to-1 female to male, and in terms of symptoms, we see higher symptoms in females to males. Next slide. Importantly, while PTSD was originally identified, focused primarily on military trauma, in the 70s, chronic rape syndrome was identified, and it was really appreciated that interpersonal trauma is one of the biggest risk factors for post-traumatic stress disorder, and we see it across the veterans, but as well as in civilians. And the highest risk for those who are both female and in at-risk communities and communities of violence. Next slide. So the work, a lot of the human work I'm going to talk about really came from my work at Emory and Grady Trauma, called the Grady Trauma Project. And this is from Grady Memorial Hospital in Atlanta, one of the largest remaining charity hospitals in the country. And this population over the last 15 years or so, we've interviewed almost 15,000 people that have all experienced trauma in one way, shape, or form, with high rates of childhood trauma, high rates of interpersonal assault, and high rates of trauma just from living in high-risk communities. We see a rate of almost 25 to 30% PTSD. We're enrolling people from general medical clinics. So these are not people seeking mental health care, though some have obviously been involved in the psychiatric system. It's really just the rates of epidemiological trauma in an at-risk community. And we see these rates whether we're looking in Atlanta, whether we're looking in Detroit, Chicago, rates where you have chronic poverty and injustice. And we see these trauma rates that are just very, very high. In addition to high rates of lifetime and childhood trauma, we also see increased depression, substance abuse, medical cardiovascular risk, criminal justice involvement, and lower overall educational achievement, all associated with trauma, both early and lifetime trauma. Importantly, this work has led to a much greater understanding of the neurobiology and genetics of PTSD. This was one of the founding cohorts of the Psychiatric Genomic Consortium for PTSD that now has almost 250,000 total samples. And it's really helped bring voice to the scientific understanding of intergenerational and interpersonal trauma in at-risk communities, and particularly the difficulty of being Black in America and the high rates of trauma that are exposed there, and how that's added to the other constraints that make it difficult for progress. Next slide. So coming back to sex differences, so on the left you'll see what we frequently see, whether it's anecdotally within people's clinics as well as in large-scale epidemiological studies, which is mood, anxiety, and stress-related disorders have much more high rates in females, whereas autism, attention deficit disorder, substance use disorders, and schizophrenia tend to have much higher rates in males. And notably, PTSD is among the highest in terms of female to male ratio, with about a 2-to-1 ratio. There are certainly non-biological differences from the components of increased sexual assault, increased interpersonal assault, certainly social components that can be explored. But interestingly, we can also see very much sex differences in rodent models. And there are clear biological differences that also are involved in this process. And so today, I'm going to be talking about, first, overall neurobiological and genetics, to give you sort of an overall primer of how we think about post-traumatic stress and trauma and fear-related disorders broadly. And then I'm going to dive into 2 different stories in some ways in which we're understanding how estrogen and progesterone modulate the fear circuitry and microcircuitry in ways that may lead to increased risks of females. And again, we often think about this on the pathology side, increased risk, but we can also think about it on the evolutionary side, that as mothers, as females that need to survive from aggressive men in the wild, if you will, evolutionarily, there may be reasons why threat responses could be evolutionarily more survival. But of course, that's teleology, and look forward to discussing more, but we'll focus on the biology for now. Next slide. A nice way of thinking about the role of the brain and the amygdala in the stress and fear response is illustrated by this classic figure by Joe Ledoux. And this person's walking through the woods, they see a stick, but they think it's a snake. And within a few tens of milliseconds, the visual sensation of the stick-like figure, the snake-like figure, goes from the eyes to the visual thalamus directly to the amygdala. So really within just a few synapses, the amygdala, which we'll talk a lot about, as the sort of fight or flight center of the brain, has been activated in this threat response. And in other sensory systems, it's similar, and in the olfactory system, smell, it's actually only 1 synapse away from the nose to the amygdala. Whereas the higher order processing from the visual thalamus to visual cortex to higher order visual cortex to higher order thought processing areas, that can take hundreds of milliseconds later. So the amygdala has activated the fight or flight survival reflex a full half second before one is consciously aware of what is happening. And what that leads to, in the next slide, please, is while it may help for survival evolutionarily, in the modern world, it more often leads to fear and threat related disorders, phobias, panic, and post-traumatic stress disorder. Next slide. The syndrome that is best understood related across species is the concept of a panic. And when somebody says they have a panic attack, they'll say, I felt dizzy, my legs gave out on me, I couldn't catch my breath, I thought I was dying, I had to get away. And we see increased heart rate and chills and hot flashes and GI distress and chest discomfort and sweating and a whole host of hardwired symptoms that are related to the panic response. And what we now know is that hardwired neural circuitry between the amygdala and downstream brainstem and other subcortical regions activate in those tens of milliseconds the fight or flight threat reflex. And we see a panic attack in a whole host of disorders. It's most associated in terms with panic disorder, but with that, we seem to not know where it's coming from. And what we think there is we've generalized the fears from the initial threats to anything maybe attacking us and how do I get away. To simple phobias, if you have a spider land on you and you're afraid of spiders, you have a threat attack and a panic attack, but you don't think of it as that. You think of it as being scared to death. And PTSD, the trauma reminders come to you, be they in a dream or be they in a real person, you have a panic attack. Next slide. And the way we can basically think about this is that the amygdala is the hardwired region that drives top-down regulation of the rapid threat reflex. And this is constantly modulated by other cortical areas, medial prefrontal areas, which I'll talk about later, as well as dorsolateral prefrontal cortical areas. Next slide. That's what works normally. In the process of PTSD and other threat-related disorders, simplistically, we can think of the amygdala being always hyperactivated, ready to respond in a threatful way, and the cortical area is not able to rein it in. And our current treatments, for example, the current medications, for example, SSRIs and other antidepressants, seem to decrease overall symptomatology and maybe decrease overall emotionality. Our dialectical behavior therapies, DBT, and cognitive behavior therapy approaches really increase top-down cortical control of the amygdala through learning and processing and extinction. What we hope, and some of what I'll show you, is that future medication approaches will really target the neural circuitry mechanisms, and particularly as we're understanding more of our disorders that really have different biological mechanisms in women and men, that we may need to sex stratify how we target these treatments. Next slide. So PTSD we can think of as a little bit deeper dive here as having a circuit that is the final common pathway of the amygdala on the upper left side, but that's constantly modulated by the hippocampus, which involves context and discrimination components, and the prefrontal cortex, which involves basically rapid decision making. Is this threat? Is it not? Do I go towards it? Do I go away? On the upper right, we see that at the time of the trauma, the conditioning experience happens. The negative event, the trauma, the fear, the pain are paired with something that was previously neutral, be it a car on a road or be it the face of a person that one was previously not afraid of. After this conditioning event that in the amygdala leads to neuroplasticity mediated by NMDA and glutamate and calcium channels and BDNF, it changes the structure of the synapses in the amygdala so that in the future, the neutral signals alone are sufficient to activate the hardwired threat response. And on the bottom left, then, that hardwired threat response that's being modulated by the prefrontal cortex and hippocampus leads to a whole host of downstream threat responses. We also know on the bottom right, from a temporal component, short-term memories that happen at the time of the trauma are initially transmitted into working memory and long-term memory through a process called consolidation. Over time, then those memories become permanent, and then we can treat them with behavioral therapies and other things through a process called extinction, which is the basis of exposure-based therapy and other talk therapies that are important. I'm going to talk mostly today about the consolidation process, that early phase when traumas go from the initial experience to the first long-term memories. Next slide. How do we study consolidation in these processes in humans? So one way of doing that, and again, we can do that in PTSD in a way we can't in almost any other disorder, is we know the initiation of the problem, which is the definition of the trauma. And so initially at the top, we began a study at Emory in Miami, where we enrolled about 500 people in the immediate aftermath of trauma after they were brought in by the ambulance or after they walked in from a whole host of everything from car accidents to interpersonal assaults and rapes. After they had been medically cleared and consented, we were able to collect a whole host of data and blood-based data in the emergency department within the first few hours of the trauma, and then follow people for a whole year with follow-up physiology and neuroimaging data to really understand the process of trauma responding. And on the bottom, this led to a very large site study of which Brigham and Women's Emergency Department, Mass General Emergency, as well as Boston City are all involved, of about, now it says 14, but it's really almost 30 sites across the country enrolling over 5,000 participants to really create the largest public database of the biological, physiological, and psychological outcomes of trauma after an event. And so I'll walk you through some of the early data. Next slide. So the first study on the next slide I'm going to show is from our work by Jenny Stevens, who is an assistant professor now at Emory. And what she showed was that these early in the aftermath of trauma, we find that the amygdala and dorsal anterior cingulate cortex, regions that we know in chronic PTSD are associated with fear regulation, are still activated very early and even predictive of those who will develop long-term trauma. So on the upper left, you see a number of graphs showing on the y-axis PTSD symptoms and on the x-axis time since trauma. And what we see is a whole bunch of different responses. Some people start with high symptoms and stay high. Some people start low and stay low, and others start high and recover over time. And what she showed is that even 2 to 3 weeks after the trauma, how active the amygdala is and the dorsal anterior cingulate, which tends to go with the amygdala, which is different than the ventral prefrontal cortex and the hippocampus, which oppose the amygdala. Those two regions, the amygdala and the DACC, they predict PTSD symptoms even a few weeks after, long before we can even officially define PTSD. And you see the correlations at both 3 months and 6 months, how active the amygdala is on the I think x-axis predicts how much PTSD symptoms people have on the y-axis. Next slide, please. We also looked with Sona Van Roysch, who's a K Award recipient at Emory, at the other regulatory components, the hippocampus, which I mentioned is involved in discrimination and extinction and suppressing the amygdala response. And she saw the opposite finding, that hippocampal activity at 3 to 4 weeks after the trauma is inversely associated with PTSD symptoms at both 3 months and 6 months. So these early brain responses to trauma can predict how one's going to do with much worse symptoms many months down the road. Next slide. A very interesting new finding from Nagar Fani, Nate Harnett here at McLean, and Sierra Carter, who's now at Georgia State University, but was a postdoc with us at Emory, has looked in this in our Grady population at the intersection of sex, race, and trauma. And specifically, they've looked at history of discrimination and self-report discrimination, how that may be related to brain activity to threat. And what they saw here is a number of findings, but most interestingly, that activity of the ventromedial brain region I was saying is involved very much in emotion regulation and amygdala regulation. That's directly associated with, on the bottom right, correlated with history of racial discrimination. And that suggests that experiences of discrimination have adverse cumulative effects on emotion regulation and fear inhibition in both these emotion regulation networks, and they also found it in visual attention networks, indicating important neurobiological pathways for race-related health disparities. So really, you're looking at the intersection of lifetime trauma, intergenerational trauma, social-emotional discrimination, and then sex differences that put this population in particular risk. Next slide. So where we are then is a complex question, but one that we do think is answerable. It's that post-traumatic stress disorder can really be among the most perfect disorders, if you will, for understanding the intersection of biology, genetics, and environment for understanding outcomes. And that sex differences are right in the middle of that component. Next slide, please. So I want to now tell two brief stories related to sex differences in biology. The first is genetics. So I mentioned that the Grady Trauma Project, it was one of the founding groups for understanding the Psychiatric Genomic Consortium. The upper right shows the first study with about 20,000 samples total from the Psychiatric Genomics Consortium. And now combined with the Million Veteran Program, we have several 100,000 that are involved. Early on, we found heritability in post-traumatic stress if one has experienced trauma that's as high as bipolar disorder and schizophrenia, which people really didn't expect. And secondly, which was interesting, is we found more genetic heritability in females than males. We still don't fully understand that, but it may suggest more biological predisposition in females versus more environmental predisposition in males is one hypothesis. On the bottom, one of the most exciting early studies is a genome-wide association study in the Million Veterans Program showing a hit, if you will, one of the top genes on chromosome 7 of the corticotropin-releasing hormone receptor, CRH receptor. Next slide, please. The CRH receptor is really the top of the hypothalamic pituitary adrenal stress axis, really the best understood combined axis for stress regulation. Next slide, please. This first story I'm going to tell you then is about a protein called PACAP, pituitary adenylate cyclase activating peptide. It's a mouthful, but it's one of the proteins that's upstream even of CRH. And so whereas CRH is involved in release of ventromedial hypothalamus projecting to pituitary, leading to ACTH release, leading to cortisol release in the adrenal gland, PACAP's one step upstream of that. It's upstream of CRF release, but it also directly acts on the pituitary and the adrenal gland. So it's really a systems biology stress response gene. What I'm going to show you is that this gene seems to be both stress responsive and sex responsive, mediating a gene by sex interaction for mediating PTSD risk. Next slide. So our first data did essentially a deep dive on the PACAP receptor gene itself. It is essentially a GWAS at a gene level. And what we find in blue and red are the p-values for gene-environment interactions, or specifically the genetic effect on PTSD symptoms. And you see one particular p-value at a polymorphism that's very high, 0.002 in females, but not at all significant in males, at a gene-wide level significant. We replicated that in another cohort showing further significance at 10 to the minus fifth for this gene-wide level analysis. We showed on the middle graph on the bottom that polymorphism was associated with more PTSD in females, but not males. And finally, this polymorphism on the bottom right in postmortem brain tissue neurons showed that risk polymorphism was associated with differential gene expression of the PACAP receptor in female brains, but not male brains. And if you look up at that little inset on the gene, you see the canonical estrogen response element. The take-home message of this is that SNP is a functional EQTL, expression-linked quantitative trait locus, that directly is involved in the regulation of the stress response gene, but it's estrogen sensitive. Next slide. These just show that the same at-risk EQTL PACAP receptor polymorphism was associated with more amygdala activation with fearful cues and with less hippocampal activation and less hippocampal amygdala functional connectivity with fearful cues. Again, bringing this gene pathway back to the circuit we've been talking about. Next slide. And finally, in animals, we know, again, one of the reasons the amygdala is so exciting to study is that it's very conserved from humans all the way down to mice and even through the most primitive vertebrates. This basic fear survival reflex works across species. Next slide. And what we found early on was that fear conditioning in mice on the upper left is associated with increased PACAP receptor expression in both amygdala and PFC, so that PACAP and its PACAP receptor is stress responsive. And on the bottom, the amount of PACAP receptor expression is stress dependent and fear dependent. And on the bottom right, it's also estrogen dependent. Donna Tefexis at University of Vermont showed that if she overrectomized rats and then replaces estrogen, the amount of both PACAP RNA and PACAP receptor RNA is estrogen sensitive in the BNST, the sexually dimorphic threat-related brain region. Next slide. The next story, so the take-home message of this is that PACAP and PACAP receptor is both estrogen sensitive and stress sensitive, and those two come together in gene regulation of the PACAP gene, which has both glucocorticoid receptors in it and estrogen response element receptors in it. And together, that's one way that estrogen and stress systems can modulate the circuit. And I'm not going to have time to show you, but at the bottom, Vadim Bolshakov at McLean and Yang Lee have shown that the amygdala modulates the BNST, a sexually dimorphic region, and through altering its inhibitory effects can then regulate anxiety and stress effects also directly through a microcircuit mechanism. Next slide. I want to wrap up then on one more story by Tennille Ramicki that really gets at hormone contraceptive effects in PTSD and really looks at the effects potentially of progestins and progesterone in the consolidation window. Next slide. A very interesting observation by Foray and Cahill in the graph in the upper left found that women who were on either, so it's a little bit complicated slide. What we're looking at is the IES on the y-axis PTSD symptoms several months after interpersonal assault or rape. And women were either already on hormone contraceptives or declined contraceptives in the emergency room, or they were given Plan B or gestrol. And what they found is that women who either were already on hormone contraceptive or were given either short-acting Plan B or longer-acting gestrol, they all had lower PTSD symptoms than those who declined and were not on hormone contraceptive. So the first thought, well, maybe they were less concerned about pregnancy and that affected their stress levels and therefore that decreased their PTSD, and that's certainly possible. But it also suggests that there might be a biological progestin-mediated effect on memory consolidation. Next slide, please. So Tennille Ramicki, who's both a Helen Hay Whitney Fellow and then a Judge Baker Fellow in our group and has really done a terrific job leading these projects, wanted to study specifically, can we replicate that question in mice? So she worked out doing hormone contraceptive work with oral contraceptives, which is not easy in mice, to really get nice control over this. Next slide. So just showing with vaginal smears, she can get normal cycling in this slide, and the next slide showing that she can get very good blockade of the HPO axis. She did 3 sets of experiments here. The first one shows that if she gave chronic in the upper left levonorgestrel, so a progestin agonist, before fear conditioning, through fear conditioning, and after testing, she saw in the upper right lower fear expression after fear conditioning, and consistently in the bottom left and bottom middle, lower fear expression in the days following fear conditioning. Next slide. She then said, if I'm trying to model that middle graph, and I don't start the levonorgestrel until after the fear conditioning. So again, in the middle, she sees normal fear conditioning. Then she still sees decreased fear expression, both overall in the fear expression test, as well as across extinction days, showing lower fear over days. And finally, in the next slide, she showed if she only gave 1 day of levonorgestrel, essentially mimicking Plan B, she can still get in the bottom right slide lower fear expression over time, thus suggesting decreased fear consolidation. Next slide. To get at this mechanism, so this is just a summary that whether she gives levonorgestrel throughout the whole process of fear acquisition and expression, or only in the consolidation window, she can get a robust deficit in fear consolidation with a progestin agonist. Next slide. To begin to get at mechanism, she then showed in the bottom left here that if she takes mice and treats them for 2 weeks with the levonorgestrel and then takes their brains and does slice physiology, that she sees significantly more inhibitory drive, GABAergic drive in the basolateral amygdala pyramidal neurons in those that have been treated with progestin, levonorgestrel. That's the only difference. And now she's working on the molecular mechanisms at a cell type specific level. Next slide, please. How we can take cell type specific sequencing across the amygdala, as well as in human males and females with PTSD, to understand the differences in estrogen receptor drive and progestin specific drive in specific cell populations in these microcircuits that can help us understand how chronic levonorgestrel progestin can lead to a decrease in fear consolidation and increase in extinction and decrease in fear expression, and potentially be a treatment not just for sexual assault survivors, but for PTSD consolidation generally, particularly in females. Next slide. So in summary, biology, meaning the intersection of sex, genetics, and the brain, interact with the environment of trauma for a healthy developing brain. PTSD is quite prevalent, 5 to 10% of the general population, but twice as prevalent in females and twice as prevalent in at-risk communities with chronic trauma. PTSD is associated with increased risk for multiple comorbidities, medical comorbidities, cardiovascular, as well as substance abuse and mental health disorders. Our understanding of neuroscience, fear, threat, and PTSD is rapidly expanding, and sex differences is critical, both in terms of trauma consolidation and in evolutionary explanations. And the two more specific data stories I told you are that estrogen modulation of the PACAP and HPA axis may be one pathway that increases risk, as well as progesterone modulation of fear in specific microcircuits in the amygdala in the aftermath of trauma. And the most important take-home message is that trauma-related disorders and PTSD can be treated. We have pretty good treatments, though we're working for better ones, and people should seek help if they need it. Final slide. Thank you so much for this great team that's done all the work I showed you, the people, and thank them along the way, particularly Tina Rameke, who's done all the recent work on the progesterone story. Thank you very much, Dean, and it's a pleasure to be at the Connor Center.
Thank you, Carrie. I'm really, excuse me, delighted that we had a chance to hear you take us through the full spectrum of understanding how the biology of the female brain developmentally, and also the endocrine basis of neural function can translate to different pathways for disease and different opportunities also for treatments. I mean, I think you highlight very importantly that, you know, this is a condition that is female dominant, post-traumatic stress. There's also sociologic factors that mean women have certain exposures and girls have certain exposures. And then the biology is different. And so it's, we always think about conditions that are predominant and also differential, that there's biological difference. So you're highlighting really key aspects of what we're doing here, that there may be specific therapies that are highly relevant for women. And until we segment and look male, female separately, we don't really afford ourselves the opportunity to get the best treatment. So I applaud you and your work and the work of others for doing that. You know, I think it's obviously PTSD is important for men and women. The veteran and soldier experience in men is hugely important, as it is for women. And you highlighted that in your talk as well. So thank you for that. We do have some questions, and I know we won't get to all of them, so thank you for being available afterward to answer some of them in the chat box. But I wanted to ask you one question that's been upvoted for your attention, which is to get your insights on the epigenetic transmission of PTSD from mother to child during pregnancy.
Yeah, so it's a very exciting and you know, somewhat disturbing. First of all, thank you, Hayden, for all those great comments. And again, it's such a pleasure to be here and to be able to collaborate with your great team and folks within the Connor Center. So there's work at different levels trying to get at this question. Some of the best work from Tracy Bale out of Maryland that's really done some amazing work on looking at intergenerational transmission of trauma via the mother, and specifically how microRNAs may be loaded into oocytes from the bloodstream, and how that may alter potential biology of the developing fetus. So I guess I'll back up one step. I guess when we think of intergenerational trauma, there's lots of different ways to break that down. The first is of course we have behavioral and observational differences. If you've been traumatized, you're probably going to parent in a different way or respond to threat in a different way that your kids will observe. Second, gestationally, certainly the mother's cortisol and other hormone levels can be transmitted. But third, then there's the question of can there be true intergenerational trauma risk? And from Tracy's work, also work by others in Europe, and work by our own group and Brian Diaz and others in males have also shown that intergenerational trauma can be transmitted epigenetically in our work through the sperm, and in Tracy's work through the oocytes. And in both cases, in sperm, we think it's in the epididymis that probably piRNAs are loaded from the bloodstream into the epididymis into developing sperm. It's less understood in oocytes, but it's clearly happening. How that microRNA then alters either DNA methylation or histone regulation, or is converted to the next step, and how that survives reprogramming is still a black box. But with in vitro fertilization and other tools like that, it's quite clear that this phenomena happens, at least at a most basic threat level. How broad it is and what the implications are remain somewhat uncertain.
And are there known differences in that in terms of whether the offspring is male or female?
There are sex differences. I'm going to get it wrong if I try to tell you what they are. Isabel Mansui is the main mother person in Switzerland and Lausanne that I was thinking of. And they've shown it's passed down, and I think it either skips or switches between male and female, and that's not quite understood. And Tracy's work definitely shows sex differences. We didn't focus very much in our work on male sperm transmission sex differences. So I don't know if it's happening there or not.
Right.
And the mechanism isn't understood yet.
Right. Thank you. So we won't get to all these questions, but questions regarding the neural circuitry implicated in PTSD. Which neural circuits do you think represent the optimal targets that we should be identifying for remodeling in female-male populations?
Yeah, and great question. From a where some of the most excitement is less about direct biological changing, because certainly most of our treatments to date just generally decrease symptoms. And the issue that makes PTSD both exciting but also makes it somewhat more complex is it's a memory, it's an engram, it's a set of life experiences. What we think is maybe the best tractable approach is combining psychotherapy that really reactivate those specific memory engrams with treatments focusing on neuroplasticity and neurorecovery to best target recovery of the memories that the people are having. That said, the process of extinction we think is medial prefrontal and dorsolateral cortex with amygdala interaction. But from a cell-based biology perspective, we may have more tractability at both the bed nucleus of the stria terminalis and the amygdala, which are very well conserved across species. And the BNST in particular is particularly sexually dimorphic. So if I had to pick one area that I think we're going to find interesting and important sex stratification differences related to fear and threat, it's going to be the BNST.
So we may have time, I think, just for one more question, and I'm going to take the interviewer's prerogative here. Sort of asked you to comment about lifespan perspective, because you talked about our warriors and soldiers and military violence. So that's at presumably late teens, early 20s, 30s, and the Grady Project goes back to much earlier age. And if you could put sort of a societal context on some of these exposures and the risks to the population, male, female, if you can segment it that way, thinking about developmentally where people are, if it's an individual trauma or some sort of repeated trauma, of course.
Yeah, it's a great question, certainly a complicated question. One thing we're increasingly appreciating from work like Marty Teicher at McLean, Caroline Ruth at Cambridge Health Alliance and Children's, and Aaron Dunn at MGH, is that the timing of the trauma in development is very critical. So whether it's fetal things experienced by the fetus versus the very early development versus the first few years versus early school years, that's hitting very different, if you will, emotional sweet spots or forgive me for blanking on the term, on really developmental sensitivity periods, I guess. And that can have very different effects. And so one thing just about childhood trauma, and then I'll comment about the rest, is that we often think about childhood trauma both on what people can remember. That's pretty much how we have it, is self-report. And yet we know that the emotional brain is maturing and we're having threat responses a full 1 to 2 years before many people have conscious memories and are fully well verbalized. So that there's stuff happening in that 1 to 2 year period where we're creating emotional memories and we don't really have declarative conscious memories of that. We don't have a clue what that does developmentally in terms of early trauma. So that's one component, the whole childhood developmental trauma component. We then know puberty is a big risk factor and changing factor for a lot of things that are very critical for everything we've talked about today. And then we know early development, late teens, early 20s is a big factor for everything, because that seems to be when a lot of the prefrontal cortex finishes developing and a lot of the reward systems finish developing. And then in terms of sex hormones, we know that postmenopausal is a different set of windows than premenopausal for other sort of hormone relevant risk factors. So I think the answers are far fewer than the questions, but at least we're starting to understand how to break these down in biologically meaningful ways.
Right. No, that's hugely important. And when you think about in this day and age with, the COVID exposures and Obviously, people happen to be the age they happen to be, and these are different. This is a mass societal trauma, but presumably people's recovery from the trauma they experience in the losses they have, health or economic, or the vulnerabilities across, particularly with our racial minorities who have been disproportionately affected. This is going to have powerful impact.
Yes.
Yeah, we have a lot more to learn. So I hope you are continuing to do this work. I think your work highlights the importance of looking in communities that have really been disproportionately burdened, and I appreciate your doing all that. So
Thanks so much to you all and for you all for highlighting it.
So I wish we could clap. I wish the audience to see the number of questions and all the attention you have, and I appreciate that you're going to be remaining on the program and joining us towards the end and digging into the Q&A box that's right there for you. We are going to shift now to the second part of our talk, so thank you, to go to the next series of presentations, which will be moderated by Dr. Joanne Manson, who's the scientific advisor to us in the Connor Center. Dr. Manson is also the chief of the Division of Preventive Medicine at Brigham and Women's Hospital, and the Michael and Lee Bell Professor of Women's Health at Harvard Medical School, and a professor of epidemiology at the Harvard T.H. Chan School of Public Health. So Joanne, thank you for moderating the next set of presentations as we go forward here.
Thank you so much, Haydeen. It's really an honor and a privilege to introduce the four speakers and to moderate the Q&A. I'm going to be introducing all four speakers at the beginning, and there will be a Q&A at the end of the fourth presentation. So please continue to submit your questions via the Q&A box. So the first speaker will be Dr. Haydeen Jaffe, who needs no introduction. And as you know, Dr. Jaffe is the executive director of the Connor Center, and she'll be sharing with you some very exciting updates from the Connor Center and a new center grant at the Connor Center. Our second speaker will be Dr. Dawn DeMayo, who will be speaking on sex, gender, and precision medicine, network medicine approaches to lung disease. And Dr. DeMayo is a 2019 Ignite awardee, a respiratory epidemiologist at the Channing Division of Network Medicine, a pulmonary critical care physician in the Department of Medicine at Brigham and Women's Hospital, and an associate professor of medicine at Harvard Medical School. Our third speaker will be Dr. Cindy Liu, who will be speaking on young adult well-being during the COVID-19 pandemic, current and potential downstream effects on mental health. And Dr. Liu is a 2018 Mary Ann Tynan Faculty Research Fellow, an assistant professor in the Department of Newborn Medicine at Harvard Medical School, and director of the Developmental Risk and Cultural Resilience Program within the Pediatric Newborn Medicine and Psychiatry at the Brigham. And our last but not least speaker will be Dr. Lee Min, who will speak on the interaction of stress and nutrition in the regulation of female reproduction. Dr. Min is an awardee of the 2019 Women's Health Interdisciplinary Stress Program of Research, or WHISPER. He's an endocrinologist in the Department of Medicine, Division of Endocrinology, Diabetes, and Hypertension at the Brigham, and an assistant professor at Harvard Medical School. So we'll now begin with Dr. Haydeen Jaffe's presentation.
Thank you, Joanne. This is really just a really exciting opportunity to let the community here know about a new grant that we have just launched this summer. So this is funded by the National Institutes of Health. This is a major initiative of the Office of Research in Women's Health, ORWH, and co-funded by the National Institute of Aging. So I'm really thrilled to say that the Connor Center was awarded one of 11 signature grants. These are 5-year awards called SCORE Awards, stands for Specialized Center of Research Excellence on Sex Difference. Our SCORE Award here, we've come up with an acronym. We're calling it ROSA. It's the Brigham Harvard ROSA Center, which stands for Reproductive Outcomes of Stress and Aging in Women. Now, to highlight what we're talking about here with this focus, which is we're talking about postmenopausal women and the effects of stress. Stress exposures over the lifespan, stress exposures early in pregnancy and even beforehand in younger adult and childhood life. And stress exposures in midlife and beyond, and how they affect the health of women as women age. If you can go to the next slide, please. So very briefly, the overview of what we're talking about here is the underlying biology is that in the brain of women and men, there's regulation of the hypothalamic pituitary gonadal axis, or HPG. That's the part of the brain that controls the reproductive axis. There's been some really important advances over the last 10 years that above that axis in the brain, there's something called candy neurons, named for the acronym of kisspeptin, neurokinin, and dynorphin. And these candy neurons, as you can see in the figure in the yellow, project both to the right in the GnRH neurons for the reproductive axis, and also discovered to project toward the left in the medial preoptic area, the temperature sensing neurons that control the primary symptom of menopause and postmenopause, which are vasomotor symptoms or hot flushes. If you can forward one. So all of these changes in the brain lead to these key, burdensome, most common outcomes in the health of women. And as women age, these symptoms can cause more or less bothersome symptoms and health concerns. The focus of our grant is on vasomotor symptoms, VMS, or hot flashes, and secondarily sleep problems. And all of these in the work that we're doing are really interrelated with changes in body fat distribution, adiposity, also cognitive changes, and cardiometabolic risks. And together these set women up for risks over time, leading to health outcomes in terms of heart health and also brain health. You can pop up one more, please. So these are really burdensome. As I said, 85% of women experience hot flashes at some point in their lives. And they persist for more than 10 years in 1/3 of women. So if you look many years after menopause in women in their mid-late 50s and beyond, still a significant proportion are experiencing them. And they affect their quality of life, and they also affect sleep and the cardiometabolic and cognitive risks. So if you pop up one more, please. Our studies will really dissect these relationships in a variety of ways by looking at how these aging outcomes are influenced by stress exposures. We know that stress is highly interrelated, as Dr. Ressler just presented, and now we're focusing on older women, how stress is linked with these many burdensome symptoms and what the brain's processing, the neural mechanisms are, connecting stress to these health outcomes. Next slide, please. So here's this really exciting and wonderful team. If you look on the top row, we've got 3 projects. This is a traditional program project grant or center grant with 3 projects and 3 cores. The 3 projects here include myself and Dr. Mann leading a clinical project. We have a population science project led by Dr. Oken and Dr. Chavarro. And then we have a preclinical animal model project led by Dr. Kaiser and Dr. Navarro. Dr. Manson and I are co-leading the leadership core. We have a sleep core led by Dr. Klerman. And we also have a career enhancement core led by Drs. Rexroad and Rich Edwards. If you can pop up one, please. Very, very briefly to highlight that the first project, which is looking at brain imaging, functional brain imaging activation studies, and also MR spectroscopy to understand GABA circuitry and brain regions involved in the processing of stress in women with and without vasomotor symptoms. If you can go up one more. The second project, as I mentioned, is a population study. This is leveraging a tremendous resource from Project Viva, which is a 20-year cohort study in which women are now in their late 40s, early 50s. And these women will be investigated cross-sectionally to look at their menopause symptom health. And these outcomes will be related to earlier life exposures and female-specific pregnancy stress and metabolic and environmental stress exposures, including racial stress exposures and other socioeconomic stress exposures. And if you can pop up one more. And the third project, which relates to the rodent model in which stress mechanisms through glucocorticoid receptor activity will be dissected in terms of kisspeptin and GABA circuitry to understand their downstream effects on the hypothalamic pituitary gonadal axis and also the thermoregulatory effects. And then very lastly, we're thrilled to say, if you pop up one more, that our Career Enhancement Core enables us with support from our institution and the NIH and broader within our real hospital-wide emphasis on sex and gender medicine, informed medicine, to support junior investigators through scholar awards, pilot awards. So we take all this science and we support further investigators so that we can have the next generation continue to do this work. So thank you very much. I'm going to let Dr. Manson take it from here.
Thank you so much, Dean, and thank you for your really outstanding leadership of the Connor Center and your help with all of these exciting new programs. Our next speaker will be Dr. Dawn DeMeo.
Thank you very much, Dr. Manson. So I'm excited to share my work that's been funded by the 2019 Ignite Award on emerging concepts in precision medicine, pulmonary disease. Next slide, please. And the reason this is important is that obstructive lung disease is the fourth leading cause of death overall, the third leading cause of death in women, and the fourth leading cause of death in men. But despite the importance of chronic lung disease to morbidity and mortality in the developed world, there's really been a lag in our approaches to understanding the pathophysiology and best practices to therapeutics. And if we think about the ways in which this may manifest differently in women, we know that women tend to be underdiagnosed and suboptimally treated with chronic obstructive lung diseases. They may present differently. They have inextricable links to poverty and socioeconomic status and stress. We know that differences in tobacco use, both tobacco use in and of itself, but how it impacts the genome, all of these issues together may impact sex differences in lung disease. And what we feel is that the integration of genetic variation in a systems biology framework may really be a seminal way forward to understanding key pathways that influence not only chronic obstructive lung disease, but other smoking-related diseases that have sex differences. And that would include cardiovascular diseases and other diseases that are complex in nature. And at our division, we believe that precision medicine and the network medicine approaches that we've developed in our group may help us innovate in this space to develop sex-specific therapeutics. And why is this the case? Well, we believe that single genes or gene pathways really don't fully characterize a disease, especially sex-specific features of complex diseases. But it's how genes interact in a network framework that matter. So we've been interested in investigating sex-specific gene regulatory networks associated with chronic lung disease, and then taking these findings and looking in silico to screen network-defined drug targets to identify sex-specific therapeutics for chronic disease of the lung. Next slide, please. So what is network medicine? So network medicine really takes us to the realm where the study of cellular disease and social networks, this idea of a network framework, all come together to impact individual diseases. And one of the intuitions of network medicine is that it's holistic. It allows us to capture nonlinear responses of complex systems, and also that it captures emergent properties that may lead to disease. And it inherently employs a systems biology framework. And if we think about sex versus gender differences and how these come together with all different sources of data we may measure in the laboratory, we can bring these data together in a network context to infer sex-specific and subject-specific gene regulatory networks for chronic lung disease. So we believe that using network-based predictions will allow us to identify key target genes. Next slide, please. So what we did, and this work was done using the development of regulatory networks to study sex differences. And this work was done by a very talented junior investigator named Camilla Lopes-Ramos over at the School of Public Health, where she took data, gene expression data from the GTEx project, which represented about 8,300 individual gene expression profiles on 29 tissues. And we looked at sex differences in gene expression. And on average, there are not a lot of differences by sex in gene expression across the compendium of human tissues. But when we bring together prior knowledge and build gene expression networks, what we find is the ways in which genes are targeted by sex differ. So it's not necessarily the end product, the expression that matters, but the road to how we get to gene expression. So we took to task to model sex differences in sputum that was provided from men and women with lung disease. And if we look at gene expression edges in these networked framework in women versus men, we saw significant differences in the genes that were targeted in the lung in women and men with lung disease. So we decided to scale this up and look at the lung and apply a concept called the connectivity map. And what the connectivity map is, it's a resource that allows us to use gene expression data to probe relationships between diseases, cell physiology, and potential therapeutics. And I won't belabor this because there are a lot of beautiful papers that describe the connectivity map, but applying an investigation of gene regulatory network edges to the connectivity map, we were able to identify sex differences in potential therapeutics. And we have already been able to identify genes based on female targeted and male targeted differences in gene regulatory networks, and hope these will give us new pathways forward to the treatment of chronic lung disease. Next slide. So I'd like to thank the Connor Center for the support for this innovative award, and my collaborators, especially my postdoctoral fellow, Daniel Morgan, who's been addressing the lung-specific networks, and Camilla Lopes-Ramos, who's been an important collaborator as we develop the methods moving forward. Thank you very much.
Thank you so much, Dr. DeMeo, for that terrific presentation and the great work that you're doing. Just a reminder to please send your questions through the Q&A box, and we're going to move to the next presentation by Dr. Cindy Liu.
Hi, thank you so much for having me. I'm a clinical and developmental psychologist focused on psychosocial stress, mental health, and disparities. And as of 6 months ago, I started to study the effects of the pandemic on young adults and new parents, specifically women who are expecting a baby or who had just had a baby. And I'm grateful for the Connor Center and the Mary Ann Tynan Faculty Fellowship, which allowed me to get this work started. Next slide. So we know that there are a couple of points in development that involve sort of major transitions. One is young adulthood, and the other is preparing to have a baby. And you know, frankly, when you go through a transition, it can take us aback as we have to adjust to a variety of new roles, not to mention the biological changes that take place during this period of time. We know that for young adults, they are trying to become independent from their parents. They're going off to school, they are graduating, and for parents, they're developing a dependent relationship with the children under their care. So as I mentioned, this is a period with major life's milestones. And it's this relatively short period where all of that takes place. And so a question that we wanted to ask was, what happens to the lives of individuals during these particular life transitions during a pandemic? What is it that we should look for and address? And as it turns out in the news, there have been a number of different articles that have been focused on these very issues, the mental health concerns that both young adults and perinatal women face. Next slide. So we decided to start 2 nationwide longitudinal studies where we enrolled participants into a 30 to 40-minute survey. In mid-April, we launched the CARE study, which is listed on the left, which refers to the COVID-19 Adult Resilience Experience study, which focuses on 18 to 30-year-old young adults. Thanks to the rapid funding mechanism from the National Science Foundation, to date, we have about 1,400 participants that have enrolled, the large majority being those who identify as women. And among our sample, we have half who are students. And then in mid-May, we launched the PEACE study, which refers to the Perinatal Experiences in COVID-19 Effects study. And to date, we've enrolled about 2,000 pregnant and postpartum mothers. And we focused on this group knowing that the major changes that have occurred in prenatal care were affecting those who are preparing to have a baby. And of course, with COVID, there have been so many limitations to seeing other people. And so those limitations, of course, impeded on postpartum support. So between these 2 studies, we've examined a range of experiences in our data so far. We've looked at relocation effects in college students. We've looked at COVID-19 related discrimination. Our survey is very thorough. What I'm going to talk about briefly is just some of the more compelling data that we have, some of the newer data that we have. Next slide. So what we know at baseline is that for young adults, there's pretty high rates of probable depression, anxiety, and PTSD. We're seeing about 2 out of 5 having probable depression, 1 out of 2 reporting probable anxiety, and about a third reporting probable PTSD. We know that mental health or having a pre-existing mental health diagnosis is a vulnerability. And so for those individuals, they're 4 to 6 times more likely to report this elevated psychiatric distress. For perinatal women, we also see at baseline really high rates. So about 1 out of 3 have elevated depression, 1 out of 5 generalized anxiety, and about 1 of 10 with PTSD. And again, here we see too that those with pre-existing mental health diagnoses were about 2 to 4 times more likely to report this elevated distress. So what happens when you control for pre-existing mental health? What do we see? What we see is that grief keeps showing up in our data. Grief is associated with lower mental health functioning, more loneliness in young adults. It's also associated with mothers who are reporting lower quality in maternal-infant bonding beyond reports of psychiatric symptoms. And so, so there's a number of concerns. Next slide. There are a number of unexpected losses that are taking place, and I'm sort of convinced that we're not necessarily recognizing that. And it's difficult for people to bring up these issues because everybody is struggling. But I do think that this grief is something that we might need to highlight. And so in terms of next steps, I'll be looking at following up with these individuals. We are applying for funding to be able to do observational studies with mothers and babies, bringing the lab into their homes, really seeing if indeed they are struggling with maternal bonding in person. And so, so there are a number of next steps with this work. Again, want to just thank you. Next slide. If you have any questions, you can contact me. I can give you more information about our data.
Thank you for sharing that really important work with us, Dr. Liu. And our final speaker will be Dr. Li Min.
Thank you so much for that nice introduction, Dr. Manson. And I'm honored to be invited to give this presentation. Next slide. So stress and undernutrition disrupt female reproduction, but the mechanisms are not known. So we know that two hormones, leptin and the glucocorticoids, both are involved in the regulation of metabolism and reproduction. So as you know, leptin is a key hormone secreted from adipose tissue that can communicate energy homeostasis to the reproductive axis. The leptin deficiency mice, as known as OBOB mice, they are obese and infertile. Glucocorticoids is a hormone that increases in response to stress. It's a negative energy balance, and it's increased during the negative energy balance and the leptin deficiency. And it is a proposed mediator of stress associated with reproductive dysfunction. So in animal study, fasting results in high corticosterone and low leptin and low gonadotropin in mice. This finding reflects a negative correlation between leptin and the glucocorticoids level during nutritional stress. And in our lab, leading by Dr. Kaiser, we found the delete of leptin receptor neuron developed a phenotype mimic OBOB mice, which means these mice are obese and infertile. So based on these findings, we proposed a hypothesis as illustrated on your right upper cartoon. There's a crosstalk between glucocorticoids and leptin to regulate reproduction under stress. And this crosstalk occur in the neuron in the hypothalamus, and these neurons express both leptin receptor and glucocorticoid receptor, we call GR. So to test this hypothesis, we generate a mouse model. It's a transgenic mouse model by leptin, by Cree and the loss technology, as illustrated in the right lower cartoon. Next slide. So the overall goal of this project is to study if crosstalk between glucocorticoids and the leptin contribute to stress-induced reproductive dysfunction in female mice. So after we have this mouse model, we characterized the phenotype of the weight and the reproduction. As you can see on your left side figure, sorry, previous slide. There's no difference between the control and the GR deletion group in terms of body weight and reproductive phenotype. In terms of H circle, age of vaginal opening, age of first uterus, time to first liter and interliter interval. Next step, we evaluate the effect of color restriction-induced stress on atrial circle in these mice. As you can see at the upper panel and the right side upper panel, during the color restriction, the weight, the mice had weight loss and followed by rapid weight gain after end of color restriction. And there's no difference between control and this GR deletion mice. On the other hand, if you look at the middle panel of the figure, the control group stopped circling shortly after color restriction. On the other hand, if you look at the lower panel, the GR deletion mice, it seems like they are more resistant to color restriction because they stop the cycle later. So this finding suggests the GR deletion mice develop resistant to color restriction-induced, stress-induced reproductive dysfunction. Next slide. So we look at the other stress on this reproductive dysfunction. We apply restraint-induced stress on this mice. We apply stress 1 hour a day for total for 10 days. And during this stress, and before, during, and after the stress, actually, we measure the body weight. As you can see in your left lower figure, you can say the weight decreased slightly. during the restraint, and there was a little weight gain after we end the restraint. But there's no difference in terms of weight change in two groups. Now, on your right upper bar graph, we look at the number of H circle in this mice. We compared the control and the GR deletion mice. As you can see, before the stress, the number of the circles in the two groups, there was no difference. And during the stress, both groups show decrease in the H circles. But if you look at two groups, you can say the control group had a significantly lower number of circles. Again, this data support restraint-induced reproductive dysfunction. The GR deletion mice has more resistance to this restraint-induced reproductive dysfunction. So our conclusions: female mice with targeted deletion of glucocorticoid receptor and leptin receptor expression neuron have normal reproductive function under basal condition in the absence of stress. However, if under stress, female mice with targeted deletion of glucocorticoid receptor and leptin receptor expression neuron display resistance to both color restriction and restraint-induced reproductive dysfunction. So this data suggests that glucocorticoid signaling in leptin receptor expression neuron play a role in the regulation of stress-induced reproductive dysfunction. Next slide. I would like to thank Dr. Ursula Kaiser, my mentor, and Dr. Rona Carroll for their advice and help, and Cindy, Grace, Heisha, Daniel, and Melissa who participated in this project, and Victor Navarro and his lab for their help. Thank you.
Exciting and very exciting work and a great presentation. So I think there may have been a problem with accessing the Q&A box, but we do have some questions. Please at this point, maybe send them through the chat box if you're having any trouble with the Q&A box. But let me begin with some of the questions that have come in. So here's a question for Dr. DeMaio. How does network medicine account for social correlates of health associated with pulmonary diseases such as asthma?
So one of the ways in which network medicine can be informative is that we can model different aspects of environmental exposures, both when we're defining the omic outcomes, so whether it's gene expression, DNA methylation, we can build our models accounting for those social correlates. This is really potentially more manifest when we look at the epigenome. So we're looking at ways to evaluate social constructs together with the epigenome and then put the epigenome in a network framework. So down the line, we hope to not only capture biologic recapitulation through networks, but the societal constructs that may be manifest through the epigenome.
Thank you so much. And we have a question from Carrie Ressler for Dr. Min, which is the effect of stress on the HPG axis only occurring through the hypothalamus, or is there evidence that other brain regions are interacting with the hypothalamus may mediate these effects?
That's a good question. So we, as Dr. Harding mentioned, previously we know HPG axis, which means hypothalamus, pituitary, and gonads axis through GnRH neuron. And now we know there's a candy neuron related to this. The short answer is we do not know, but I think there might be involvement because we do see GR in existence in like a hippocampus, but I think that should be some area we can do research in the future to look at this link and you know, to say if like glucocorticoids at a higher level to impact on reproduction.
Thank you, Dr. Min.
Yeah.
And a question for Dr. Liu. How can the findings from your study on maternal stress exposure be translated at the clinical level? Is there any particular target intervention that you think should be developed or implemented?
That's a great question. So I do think that CBT that's focused on the loss experiences may be really critical during this time. There are a lot of unmet expectations, and what I didn't get into were the qualitative responses. The large majority of respondents actually said a bit more about, they elaborated a bit more, and what we saw consistently was just that they were losing experiences that they expected, whether it was graduations, weddings that were broken off, and for mothers, it was not being able to celebrate the birth of their baby the way that they thought they would. And this was, it was actually very devastating to read it, but very much in line with what we're seeing in the data, that this, perseverating on that, we want to validate those experiences, but at the same time, we want to be able to help people still function in light of the losses that have occurred. So I believe that CBT might be useful if it were adapted in that way.
Fantastic. And another question for Dr. DeMayo. Let me find this. Do males and females respond differently to inhaled steroids given many genes that are regulated by both glucocorticoid receptors and estrogen receptors?
Yeah, that's a terrific question as well. And as we look at the pharmacoepigenetics specifically, we've been interested in studying this because our prior work has suggested epigenetic differences in response to systemic steroids. So we're going to be looking at both sex differences in the lung for inhaled as well as systemic steroids. We haven't finished that work yet, but we believe they probably do have differences.
Dawn, have you seen much of an effect of estrogen therapy in postmenopausal women on any of these respiratory outcomes?
So in my own work, in one of our biggest studies, we actually haven't collected those data in a detailed fashion, but it is a direction going forward as many of our COPD patients are postmenopausal, but we haven't been able to look at the genomic interactions yet in the network framework.
Yeah, there was a suggestion from the Women's Health Initiative that estrogen might have a favorable effect, but the statistical power was a little low. I have a question here for Dr. Liu. Have you found any racial ethnic differences in your CARE study?
We have actually. So Asian Americans relative to whites were less likely to report depression and anxiety. And we also saw that Latinos also reported less anxiety relative to whites. In some of the discrimination analyses, we do see that COVID-related discrimination was associated with higher PTSD levels in Asian Americans.
Interesting. Continue to send your questions through probably the chat room at this point. I have a quick question for Dr. Min. You had discussed the effects of stress on the female reproductive system in rodents, but how much do you think that the findings in female mice mimic the stress response in humans? And also, do you have any data on the effects of stress on the male reproductive system?
Yeah, that's a good question. So we do know the human being, for example, like the female with anorexia nervosa, that's kind of stress. This kind of women, they always have hypercholesterolemia slightly, and they have a disrupted reproductive function. And I think there's a correlation between our study and the human phenotype as well. So, in terms of sorry, I'm sorry. What is the next question?
In males.
Okay, yeah, we do have some fun studies on the male, and there's difference. I think a male, for some reason, is more resistant to this stress. That's we have this data, but we haven't had a good analysis.
Or this may be one area where males are actually less well studied, especially in laboratory animals.
Yeah.
Okay, we have only 5 minutes left, but we have another question here for Dr. DeMeo. And as a pulmonary expert, what do you think are the primary sex-based differences in respiratory failure in COVID-19? All of these differences that we're seeing in terms of men having the more severe COVID illness, need for mechanical ventilation. What would you feel might be underlying cause for these sex-related differences?
Yeah, I think this is a really important and timely issue. I think it's multifactorial. I think we're going to be able to identify the genomic differences. These are under study now, whether it's, again, going to be differences in how the ACE2 receptors targeted or something along those pathways. There are important differences related to immune responses, related to viruses. Sabra Klein has published some beautiful work in this space. So I think it's going to be some combination of genetics, genomics, epigenetics, innate immune responses. I think it's a really mandatory issue for study as we see these important confluence of race differences and sex differences as the COVID pandemic persists. So I'm not sure I can say what the primary issue is going to be. I think it's going to be a compendium of issues driving the sex differences for sure.
And what about in terms of response to vaccinations? Do you expect to see sex differences? There's been some evidence that women may have more robust responses to vaccination, but are you expecting to see much of a difference in terms of the response?
Yeah, I would be honest to say vaccine response is a little outside my expertise, but you know, I think, again, this is where we need to look at the literature on other emerging viruses and the patterns of sex differences so we're prepared. But I can't say I even want to speculate before we even how the vaccines are going to be developed and deployed.
Okay. And let me ask you, just staying with you for a minute, Dr. DeMeo, it's known that smoking is one of the risk factors for adverse outcomes, respiratory outcomes. Is there any evidence yet on whether vaping is also a risk factor? Has that been looked at?
Gosh, that's a really timely issue. And if we extend what we know about lung differences related to smoking, this is an emerging exposure that we need to be very mindful of. We know that there are differences starting at the level of the epigenome related to smoking, and we have ongoing studies to look at vaping differences. So I think in our college and younger individuals, I do think we need to be mindful of the sex differences related to vaping and how that may impact COVID susceptibility. Again, this is all emerging literature, but I do think we're going to see important sex differences related to those exposures.
Thank you. Well, we'll try to end right on time. I think we've made it through most of the questions. If not, we'll try to get back to you with responses. But I want to thank the speakers for absolutely wonderful presentations and such exciting research. And I want to thank the audience for your engagement and great questions that you submitted. And also thank you to the Connor Center and all of the supporters of the work at the Connor Center. And we wish everyone the very best and stay healthy and safe. Okay, bye now.
Joanne,
Are you going to make some comments?
The appreciation to you for moderating, to Carrie for kicking us off with that wonderful presentation, and to Lee and Cindy and Dawn for taking this great work forward. We're really thrilled to be able to support the work you do. For those of you who are attending, stay tuned for more. You know, we continue to have additional virtual programming, and this will include in late October a podcast highlighting some of the binging some of the work and discussion with Dr. Ressler and Dr. Liu. And that'll be released. You'll be able to get access to that if you follow us on Twitter and also see us on our website. And also after that, we will be hosting a webinar in November 13th, which will be focused on some additional topics related to telemedicine and its impact on the health of women. So we look forward to engaging you in further discussion over time. So thank you all very much and be well.